So in this lab, we are going to talk about protozoans. And in addition to watching this video and part 2, which continues on talking about more protozoans, you should also watch the Discovery Channel video that I've posted the link for on Canvas. It is called Eating Us Alive. And so 2 of the extra credit assignments that I have assigned for you would be to use the protozoan table. So basically, it's a table that I've provided on Canvas, and your job is to fill that in and then submit that for extra credit while watching these videos. And then for the Parasites Eating Us Alive video, there are also questions that go along with that video. And so if you answer those questions and submit them on Canvas, that is another way for you to get extra credit for this material. So first, let's start by discussing a little bit about protozoans. You might recall that we did talk a little bit about protozoans back in chapter 1, but we need to review to talk about what are the overall characteristics that protozoans have in common. So the first characteristic is that they are eukaryotic. And remember what that means is that they have a membrane-bound nucleus. So they have a membrane-bound nucleus which surrounds its DNA, and that makes it different than, let's say, bacteria, which we've been talking a lot about. So protozoans are going to be eukaryotic. Remember that protozoans are more animal-like, and what that means is that they lack cell walls. So just like animal cells lack cell walls, so do protozoans. Protozoans have stages that contain structures that are like a cell wall, but it's not a characteristic cell wall. Again, they're animal-like. They are usually heterotrophic, which means they have to consume their food in order to survive. They inhabit, typically their environments would be soil and water. You're going to see that a lot of them are found either in soil or water. They are unicellular, which remember means that they exist as one single cell. So they are single-celled. And in terms of their method of motility, remember that motility is movement. And so they have several methods through which they can move. One example would be through what we call pseudopods. And what you can see in this picture here is you can see this cell is producing these pseudopods. So here are these extensions that they're sending out. And those extensions will allow them to engulf either food particles or to move through what's called an amoeboid type movement. It's a crawling motion. Amoeba do this, for example. You might recall that I showed you the video that had the white blood cell chasing the bacteria. Our white blood cells also move using pseudopods. And those pseudopods are basically actin filaments working together with the motor protein myosin. And when they work together and they contract, it causes this, when this contraction occurs, it causes this what we call cytoplasmic streaming. And that is that it forces the cytoplasm to be pushed out, which leads to those extensions. And so some of the protozoans that we're going to talk about move using pseudopods. Some move using flagella, that long tail-like whip extension. Some move by cilia. They have lots of little tiny hair-like extensions covering the surface of the cell. And then some are going to be non-modal. They're not able to move. So when we talk about protozoans, there are typically 2 stages to the life cycle. The first stage is the trophozoite, or what we commonly would call the troph stage. And the troph stage is the feeding and growing stage. And generally, this stage feeds on bacteria and small particulate animals. Again, remember that most protozoans are heterotrophic. They have to consume their food. The cyst is the protective stage that allows for the organism to survive when food, moisture, or oxygen are lacking, when temperatures are not suitable, or when toxic chemicals are present. It also allows for the organism to survive outside of the host. And so one of the things that you're going to see is that if the organism has both stages, if they have a cyst and a troph stage, usually it's the cyst stage that is going to be infectious, meaning that's the stage that's going to cause disease. And that's because that stage, that cyst, is very hardy. It's very resistant to the elements, and it's also very resistant to stomach acid. And so as a result, that's the stage of the life cycle that if you happen to ingest it, that stage is going to be the one that causes disease. Now, for each protozoan, there are several things that you want to know. And again, these are indicated in the table, meaning I put columns in there for you to write this information for each one of these components. So for each protozoan, you want to know what is the disease that it causes. So what is the name of the disease? What is the mode of transmission? So how do you get the disease? What stage is infective, meaning which stage do you have to come in contact with in order to become infected with this organism? You want to know the method of motility, so how do they move? You want to know symptoms, so what are the symptoms of the disease? What are the treatments? What are the methods of diagnosis? So how is this disease diagnosed? And then what stages are there in the life cycle? So do they have a cyst? Do they have a troph? Do they have other stages? So you want to know what are the stages in the life cycle. So the first one that we're going to talk about is Entamoeba histolytica. And Entamoeba histolytica is the cause of amoebic dysentery. So when we write the disease, the name of the disease, amoebic dysentery, what's also called ambiasis. Now, dysentery, if you've ever played the game Oregon Trail, you might have seen your character die of dysentery. And you might not recall what exactly that means. But dysentery is basically a condition where you get inflammation of the colon. And when you get inflammation of the colon because of this organism, it causes severe diarrhea. And so dysentery is going to be when you get this severe diarrhea. And so Entamoeba histolytica causes amoebic dysentery. It causes ambiasis, and again, that's going to lead to diarrhea. In terms of method of motility, it moves using pseudopods. So its method of motility is that it's going to produce pseudopods. The stages that you need to know for the exam is you need to know the cyst and you need to know the trophozoite. What you're looking for to identify Entamoeba histolytica is you're looking for these bullseye nuclei. So what that means is it looks like this, almost like a little eye. And so if you look, here's one bullseye nuclei, here's two. So the cyst is typically going to have up to 4 nuclei. And that 4 nuclei we call a quadranucleate cyst. The quadranucleated cyst is the infectious stage, meaning that's the one that if you ingest, it's going to lead to infections. So the quadranucleated cyst is the infectious stage. Again, you are looking for those bullseye nuclei. The cell is typically round in shape. If I compare that to the trophozoite has 1 nucleus. And again, it's that bullseye-shaped nucleus. And notice that the troph has a very abnormal shape. So it's not always going to have the same shape that you're seeing here. Because remember that I just said that Entamoeba moves using pseudopods. So that means that the cell shape changes in response to the organism moving. So let's talk about now the next part of our table. is going to be the infectious stage and the mode of transmission. So again, I said that the quadrinucleated cyst is the infectious stage. It's the one that causes the infection. And the quadrinucleated cyst, the way that you get it is that it's transmitted by fecal oral route. So let me underline this. So it's fecal oral transmission. And that is that you get this by drinking contaminated water or eating contaminated food. So one of the things that you're going to see is if somebody is infected with this organism, right? If somebody is infected with this organism in bowel movements, they are going to shed cysts. If, let's say, somebody went to the bathroom in a stream and then you happen to drink that water, well, now you're ingesting those cysts. And if it's the quadrinucleated cyst, it's going to cause an infection. And so this is fecal oral transmission. So what happens is that the cyst is ingested. So notice we get ingestion. And it's going to undergo what's called excisement, meaning that it hatches in the small intestine. So the cyst is able to withstand the stomach acid. And so the cyst is going to pass from the stomach into the small intestine, where it's going to undergo excisement. Now, in the intestine is less acidic, right? The pH becomes neutralized as the food that you ingest moves from your stomach to your intestine. And what's going to happen is when it undergoes excisement, it's going to go through mitosis. So it's going to divide, and it's going to give rise to 8 trophs. So it's going to produce 8 trophs per cyst. And so those trophs are then going to be found in the intestine. So infectious stage, again, the quadrinucleated cyst, mode of transmission, fecal oral transmission. So drinking contaminated water or eating fecally contaminated food, for example. So let's move on and then talk about symptoms. So let's talk about what are the symptoms of the disease. So let me go ahead and change my ink here. So in terms of symptoms, symptoms are going to be bloody diarrhea, and the reason that Entamoeba histolytica can cause bloody diarrhea is because the trophs, one of the things that they can eat as food is a red blood cell. So this little dot that you're seeing in this diagram is actually a red blood cell that this troph has consumed. And so what happens is when the troph is in the intestines, it can actually penetrate. It can penetrate the lining of the intestine. So penetrates the lining of the intestine and causes ulcerations. Which is what is going to lead to that bloody diarrhea. And so that, again, is going to lead to our disease, which is going to be amoebic dysentery or ambiasis. Some people are asymptomatic carriers. And so only about 10 to 20% show, so only 10 to 20% of infected people show symptoms. So there is a large percentage of people that do not show symptoms, but still are infected with the organism and can still transmit the disease. We're seeing this right now with what's going on with the coronavirus, right? And that is that people might be asymptomatic. Either they're not going to get symptoms at all, or it's still during the incubation period, but they're still able to transmit the disease. And so same thing with Entamoeba histolytica. Only a small percentage of people who are infected with Entamoeba actually show symptoms, about 10 to 20%. So in terms of treatment, the treatment is going to be something called metronidazole. Otherwise known as Flagyl. And I'll talk more about that in a minute. So that is your treatment. So we'll come back to this and how this drug works and side effects. Now, in terms of diagnosis and additional notes, so other things that you should know about this particular organism, and that is that the troph is going to be in the intestine, right? And so in the intestine, that troph is going to use red blood cells for food, for example. It's going to penetrate the lining of the intestine, and it causes that ulceration or that bleeding. When the trophs make their way from the small intestine to the large intestine, the water is sucked out of the intestines, meaning the body is going to reabsorb that water, and they will go back into their cyst form. So the trophs will go back to the cysts and shed in feces as cysts. So the trophozoa is going to go back to being a cyst, and then the cyst is going to be the infectious stage. Now, hundreds of millions of cysts come out in a bowel movement. So a person can transmit this fairly easy if they are infected, because lots and lots of these cysts come out, and then if somebody else happens to ingest this, they are also going to get amoebic dysentery. So in terms of diagnosis, so notice that the cysts are going to be shed in the feces. So the way that this is diagnosed is looking at a stool sample to look for cysts or trophs. So the way that this is diagnosed is, again, it's going to be by stool sample. So looking for the presence of these cysts or these trophs in the stool sample. Now, the people who are really at risk for this, almost like most diseases that we know, is that people who are immunocompromised are more at risk. So in immunocompromised, what happens is, the problem is that the parasite is able to burrow through the small intestine. So goes through small intestine, goes into the brain, and this causes amoebic encephalitis, which basically is swelling of the brain due to this parasite, and ultimately it leads to death. And so this can be a very severe disease in the case of patients who are immunocompromised. Where is this organism or where is this disease problematic? This happens in Africa, it happens in Russia, Mexico, and parts of South America. And so we don't see this as much in the US, but it can occur. And in terms of Entamoeba coli, there's one that's related to it that's called Entamoeba coli. And so the reason I wanted to bring that up is because remember when we talked about whenever you talk about an organism for the first time, that it's important that you write out the genus and the species? Well, here's an example of that, right? Entamoeba coli, if you abbreviate that, is also E. coli. So that again illustrates the reason why when you talk about an organism for the first time, you must write out genus and species, and then you can abbreviate it in subsequent times with the genus being abbreviated. So the next organism that we're going to talk about is going to be Balantidium coli. Balantidium coli causes the disease Balantidiasis. So here we go, Balantidiasis. And again, Balantidiasis, like Entamoeba Histolytica or like ambiasis, is also going to cause dysentery. We'll talk more about that in a minute. Method of motility. Method of motility is cilia. It's the only ciliated pathogen that causes human disease. You might recall that prokaryotic cells do not have cilia. They can have flagella for motility, but they do not have cilia. So this is going to be limited to protozoans, for example. And this is the only ciliated pathogen that causes human disease. In terms of stages to know and to identify on the practicum, we have the cyst and the troph. So you need to know both and be able to identify both. And what you're looking for in this is a kidney-shaped nuclei. So we have this kind of kidney-shaped nuclei, and the cyst tends to be more circular, whereas the troph still has that nucleus. You can see it darker stained here. But the troph, the cell is more oval in shape. It's not quite as round as the cyst. And the other thing that you'll notice when you look at the troph is that it has, if you look carefully, it has little tiny hair-like projections, which are the cilia. And so you need to identify both the cyst and the troph on the exam. Now, in terms of mode of transmission, so based on what we've learned so far, would you guess that the cyst or the troph is the infectious stage? And so it's going to be the cyst. The cyst is infectious. And the way that this is transmitted, again, is fecal oral transmission. So eating contaminated foods, or drinking contaminated water. And so again, typically it's sewage containing water or sewage containing food. So meaning, again, fecal matter. So if feces, if somebody is infected with this organism, and that organism happens to get on the food or is in the water, and you happen to ingest this organism, you are going to end up with balantidiasis. Just like for Entamoeba, when you ingest the cyst, right? So you ingest the cyst, and it has this cell wall-like structure, something called a pellicle. And when you ingest the cyst is going to survive the stomach acid. And again, like Entamoeba, it's going to undergo excisement in the intestines. So let me change my ink here. In intestines. And so that is then going to give rise to the troph. Now, one of the other things that I want to mention about the way that this is infectious is that in some countries, in poorer nations, having access to fertilizer is not readily available. And so one of the things that in some countries happens is that they will use human feces as fertilizer. They'll use it as fertilizer to provide nutrients for crops to grow. Now, the problem with this is that if the fecal matter comes from somebody who is infected and those cysts come out in the feces and now they're on those crops, and let's say you eat those fruits or vegetables that have been contaminated by the fecal matter, and let's say you don't wash it thoroughly, or maybe the water sample that you're drinking from is contaminated by fecal matter. These are all ways that you can get the disease, as well as having poor hygiene habits or being around somebody who has poor hygiene habits. And what that means is that, let's say somebody is infected and they go to the bathroom and they don't wash their hands. Well, if they're now preparing your food and they have cysts from the feces on their hands and they prepare your food, well, then you could end up with Balantidium as well. So there's many ways that you can consume fecally contaminated food or water. So again, using human feces as fertilizer is a big problem. Water that's been contaminated either by sewage or by somebody having a bowel movement in it, etc., would all be ways that you could get Balantidium. So now let's talk about the symptoms of the disease. So I'm going to change my ink here. So symptoms. Often asymptomatic. But can have bloody diarrhea. Okay, they can have bloody diarrhea. And again, because it can penetrate the lining of the intestine, so this also can penetrate. So, but can have bloody diarrhea, dysentery. So again, that inflammation of the colon. Let's see, weight loss. Stomach pain. Etc. And so again, dysentery is that inflammation of the colon, and as a result, it leads to bloody diarrhea. The diarrhea can contain blood and mucus, and in severe cases, untreated dysentery can be fatal. If you recall back to when we looked at the top causes of death in the world, you might recall that one of the big causes, infectious cause of death in the world, is actually diarrheal diseases, right? And so you have to realize that in other countries, this is a lot more problematic, and the dysentery that goes along with it can be quite severe. In terms of treatment, so treatment is again going to be metronidazole. Again, otherwise known as Flagyl. And I will talk more about this in just a minute. And so in terms of diagnosis, so just like Entamoeba, the trophozoite is in the intestine, and the trophozoite will go back to being a cyst. And again, that cyst is going to be shed in the feces. So based on the fact that cysts are shed in feces, what might you guess, or even trophs could be shed in feces, what might you guess is the method of diagnosis? How do you think that this disease is going to be diagnosed? And so hopefully now that you've figured out that the method of diagnosis, stool sample. So looking for trophs or cysts in stool sample. So again, method of diagnosis is going to be a stool sample. And so one of the things that you really want to think about when you're studying these protozoans is to try and see the things that they have in common. And that is Balantidium and Entamoeba Histolytica have a lot in common. They have a lot in common. And that's because they're both transmitted by fecal-oral transmission. They both have the cyst stage as being infectious. That fecal-oral contamination is by eating contaminated food or water. The cysts undergo excisment in the intestines. The troph is found in the intestines, and because this also burrows through the intestines, the symptoms are very similar. So again, that bloody diarrhea, treatments are the same, diagnosis is the same. One of the big differences would be the method of motility. Entamoeba histolytica moves by pseudopods. Balantidium moves by cilia. When we look at Balantidium coli, it's not as severe of a disease typically as Entamoeba histolytica. And Balantidium coli infections in humans is rare in the US. It's found throughout the world, but is most prevalent in tropical and subtropical regions and in developing countries. Because pigs are an animal reservoir of this organism, human infections occur more frequently in areas where pigs are raised, especially if good hygiene is not practiced. So again, it's not as problematic in the US, but in other parts of the world, this would be more of a problem. So the next organism that we're going to talk about is Giardia lamblia, otherwise known as Giardia intestinalis. And the disease that Giardia causes is called giardiasis. And there are other names for this as well that you should know. It's also referred to as backpacker's diarrhea or beaver's fever, which likely tells you something about the disease. And we'll talk more about that in just a minute. In terms of its method of motility is flagella. And so coming off of the trophozoite are 4 pairs of flagella. And so it's a little bit tricky to see in this diagram, but you would see the flagella, and that is its method of motility. In terms of the stages to know and to identify on the exam, you are looking for the cyst. So the cyst is one stage. And what you're looking for in the cyst is it has this protein structure, this little line down the middle, that's referred to as the median body. And so what you'll usually see is you'll see that little line down the middle, and around it, you're going to see up to 4 nuclei. So it's this kind of little oval or football-shaped cyst. The troph is another stage that you need to identify. And the troph has 2 nuclei, and the nuclei are bullseye shaped. So think of the nuclei look like entamoeba. And if you look at this, it kind of reminds me, I think of the troph as having a little owl face. It looks like it has 2 eyes looking at you, and it's checking you out. So I remember this one, Giardia, as the owl. So it looks like a little owl face. It has its 2 nuclei and its 4 pair of flagella. Now, in terms of the infectious stage and the mode of transmittance, so notice that one of the diseases that this causes, or that it's called beaver's fever. Beavers are carriers and shed cysts in the water during a bowel movement. So beavers are carriers, right? And so if they're infected with this organism, they will shed the cysts into the water during a bowel movement. Now notice the second part of this, backpackers' diarrhea. And what that means is that backpackers drink untreated wilderness water. And so the way that this is transmitted is the cyst is infectious. Again, remember that I said that if they have a cyst and a troph stage, typically it's the cyst stage that's going to be infectious. And its method of its method of transmission is ingestion of contaminated food or water. So again, this is also fecal oral transmission. So it could be transmitted through human fecal matter. It could be transmitted through beaver fecal matter. And so again, backpackers' diarrhea is because backpackers will go out, they'll collect water from, let's say, a stream. And one of the ways that sometimes people will try to clean water from the wild is that they will use chlorination taps. So normally in our water supply, we have chlorine in there. And the chlorine is used to treat the water, basically to help get rid of microbes. However, Giardia cysts are notoriously resistant to chlorine. So the cysts are typically found in about 18% of water supplies that are tested. And that's because they're resistant to chlorination in the water. And so the way that you would actually need to treat your water, so if you're ever backpacking, this is good to know. The way that you would need to treat your water to prevent getting giardiasis is that the cyst would either need to be removed by filtration, so using a filter apparatus to purify the water, or by boiling the water. If you boil the water, that will kill the cyst and make it so that infection does not occur. But adding a chlorine tablet is not going to work because this organism is resistant to chlorination. So swallowing as few as about 10 cysts might cause someone to become ill. So it's not like you have to ingest a lot of these. Even a small amount can cause an infection. The cysts are very hardy and can survive several months in cold water. So even outside the body, the cysts are able to survive in that cold water. Now, again, you get the disease when you ingest the cyst. So you ingest the cyst, and just like the other two that we've already talked about, excisement happens in small intestine. So it's going to hatch and it's going to give rise, that cyst is going to divide into 2 trophs. So each cyst gives rise to 2 trophs. And the troph has a sucking disc on its ventral side, so its belly side, and it attaches to the intestines and it eats bacteria. Now, one of the differences between Giardia and the other two, so Entamoeba and Balantidium, is that for this one, does not penetrate the intestine. So no bloody diarrhea. You're going to see when we write the symptoms, it does cause diarrheal disease. However, it does not cause bloody diarrhea because it can't penetrate the lining of the epithelium. So it's going to lead to watery diarrhea. And so this would be our troph. Now, in terms of symptoms, so let's talk about our symptoms. So acute giardiasis develops after an incubation period of about 1 to 14 days. So typically this is going to happen on average at about 7 days. So about 7 days after you ingest this is when you would start to see symptoms of giardiasis. The symptoms usually last between 1 to 3 weeks. And in otherwise healthy people, symptoms of giardiasis could last 2 to 6 weeks. Now, what are these symptoms? And so our symptoms are going to be, symptoms. So our symptoms are going to be watery diarrhea, sometimes for weeks. So this can persist for a long time. Fatigue, so being tired, nausea, flatulence, meaning gas, weakness, weight loss, and abdominal cramps. So again, you're going to notice that the symptoms of giardiasis, balantidiasis, and ambiasis are all very similar. One of the differences, again, being that Giardia does not cause bloody diarrhea, but instead causes watery diarrhea. Now, again, these symptoms can last between 1 to 3 weeks or up to 2 to 6. In severe infections, so in cases where the patient has a lot of these, one of the ways that it can cause weight loss is that it can occupy so much of the intestinal wall that it prevents food absorption. And so in chronic giardiasis, the symptoms are recurrent and malabsorption and debilitation can occur. So it can be very severe if it occupies a big part of the abdominal wall. Now, 7% of the population are healthy carriers. So a portion of people can be asymptomatic and they can carry it and they can transmit this organism. So those are our symptoms. In terms of our treatment, the treatment is metronidazole again. Otherwise known as Flagyl. And again, in the next slide, we will talk more about this drug. Now, in terms of diagnosis, right? Based on what you've learned so far for the first two, and based on the similarities that they have to Giardia, what might you guess would be the way that this is diagnosed? And so if you said stool sample, you are correct. So diagnosis So diagnosis is stool sample to identify cysts or trophs. So again, you would use a stool sample and you would look for the presence of cysts or trophs. Because again, just like the others, as the trophozoite passes through the colon, it's going to go back to forming a multinucleated cyst. And the multinucleated cysts are then shed in the feces and can produce infection of a new host upon ingestion. Again, remember that the cysts are very hardy and can survive for several months in cold water. Cysts are instantly infectious once they leave the host through the feces, and an infected person might shed between 1 to 10 billion cysts daily in their feces, and this can last for several months. So this can be highly infectious. And so that is everything you need to know for Giardia lamblia. So the last one that we're going to talk about is Trichomonas vaginalis. Now, Trichomonas vaginalis causes trichomoniasis. Trichomoniasis is a STI, so sexually transmitted infection. So sexually transmitted infection. Most common curable STD in the US. So most common curable sexually transmitted disease in the US. Its method of motility is going to be flagella. So it has 4 flagella, which it uses for motility. The stage that you need to know for the exam is going to be our trophozoite stage. There is no cyst stage. There is no cyst stage. So there's only a troph stage. And so what you're looking for is you're looking for this large nucleus, and you're looking for 4 flagella coming out. The way that this reminds me, I think of Trichomonas as the nucleus kind of looks like a brain, and the flagella remind me of like neurons. So it's like a brain and it has these neurons coming out. And so that's what you're looking for Trichomonas. So Trichomonas has only a troph stage, but it does not have a cyst stage. So you can probably guess what the infectious stage is, right? The infectious stage is going to be the troph. So this is infectious. Now, is it likely, because there's no cyst stage, is this likely to be fecal oral transmission? And the answer is no, because in all the first 3 examples, the cyst was the stage that can be ingested and then cause disease. This doesn't have a cyst. It only has a troph. So again, the way that it's transmitted is through sex. So through sexual contact with an infected person. And so having sex with somebody who is infected can lead to infection in the partner as well. So that's the way that it's transmitted. Very simple. Now, when we look at symptoms, so in the US, an estimated 3.7 million people have the infection. So that's a lot of people that are actually infected with Trichomonas. But only about 30% develop any symptoms. So many people, so we can put for symptoms, many people are asymptomatic. And that's important because if you think about it, having unprotected sex with somebody, they might not have any symptoms of the disease, but they might be able to transmit it to you because they don't know that they're infected and therefore can pass it to you. So only about 30% of people develop any symptoms of trichomoniasis. Now, when trichomoniasis does cause symptoms, they can range from mild irritation to severe inflammation. And so some people with symptoms will get them within 5 to 28 days after being infected. So it could be as much as a month after you've had sexual contact with somebody who's infected. But others do not develop symptoms until much later. And in some cases, symptoms can come and go. So a person might have symptoms for a while, the symptoms might resolve, they might reappear, etc. Now, the symptoms for males and females for trichomoniasis, because it's a sexually transmitted disease, the symptoms between males and females can be a little bit different. So in terms of women with trichomoniasis, so let me change my ink here. So women will experience what's called vulvovaginitis. So that is an inflammation of the vulva or parts of the vagina. So symptoms are going to be itching, burning, redness, right? Redness or soreness of the genitals. Discomfort with urination. You can get a change in the vaginal discharge. And so notice that it says greenish, yellow, frothy, foul odor discharge. So that discharge that can be produced when somebody is infected might have this kind of unusually fishy, funky smell. The discharge might be greenish yellow. It could be clear or white as well. It doesn't have to be yellow or green. But it's often going to have a very foul smell associated with it. In terms of males, male symptoms, obviously they don't have vagina, so their symptoms are going to be a little bit different. Males are going to have itching, or, let me erase that comma, itching or irritation inside the penis. Burning after urination. So burning after urination or ejaculation. Or discharge from the penis. So they might have additional fluid that comes out of the head of the penis. And so those are the symptoms for men. Now, those are my symptoms. So now I need the treatment. And so notice that the treatment is the metronidazole or the Flagyl. And so this takes basically, you need to take this for a 2-week period. And in terms of to avoid becoming reinfected, if you happen to test positive for Trichomonas, you would need to start the Flagyl, and both sexual partners would need to get treated and would need to wait 7 days after treatment until resuming having sex again. Because if you don't wait and you keep having intercourse, you're just going to keep passing the organism back and forth. And so both partners need to get treated, and you need to abstain from sex for about 7 to 10 days before resuming sex so that you prevent getting reinfected. Without treatment, the infection can last for months or even years. So again, somebody could potentially be a carrier of this and may not have symptoms, but they actually can transmit it to their partner. Now, other interesting facts about the flagell or the metronidazole is that flagell has what's called an anti-abuse property. And what that means is that you can't drink while you're on this medication or you will get very sick. It will make you vomit. It will make you not feel good. And so this one is not just like, oh, don't drink because it's not going to make it as effective. It actually will make you very sick if you consume alcohol while taking flagell. Another thing that's interesting about flagell is a side effect that can occur is this picture down on the bottom right here. You can get what's called a black hairy tongue. Now, it's not actually hair. The black hair looks or is a result of a buildup of dead skin cells on the numerous tiny projections or papillae on the surface of the tongue that contains the taste buds. These papillae, which are larger than normal in this side effect, can easily trap and be stained by tobacco, food, and other substances such as bacteria or yeast. And so things get trapped in those papillae. They can give a foul smell. People who have this might have halitosis or bad breath. And they get the side effect known as black hairy tongue. Now, interestingly, or not interestingly, hopefully you never have this happen, but in some cases, that side effect of flagell have been known to be permanent. It's not always. Sometimes when you stop taking the drug, that symptom will disappear, that side effect of the drug will disappear, but there have been cases where that side effect doesn't resolve. It ends up being permanent. And so this is just a side effect that can occur when you take flagell. Now, in terms of diagnosis, so let me change my ink again. So diagnosis. So do you think it's a stool sample? No, right? Because this is not in the gastrointestinal tract. It's not in the GI tract. So it's not going to be by a stool sample. It's typically not based on symptoms alone, right? Because other conditions can mimic symptoms similar to this. So it's not just by symptoms alone. So typically the way that this is going to be diagnosed is through molecular. Excuse that M there. Pen got a little crazy. So molecular testing. So doing a variety of molecular tests and a wet mount of discharge fluid to look for the troph.